Hi, everyone. Welcome to the Ethel Browne Harvey Postdoctoral Seminar Series. My name is Austin Seroka, and I'm a postdoc at the Fred Hutchinson Cancer Center in Seattle, Washington. And I'll be moderating today along with my co-host, Heather Dingwall. We're really excited to showcase the work of outstanding postdoc members of the SDB. And today we'll be hearing two fantastic talks from Olya Yarychkivska from the Rockefeller University and Anthony Galenza from Stanford. Each of the talks will be 20 minutes, followed by 10 minutes of Q&A. So please utilize the Zoom Q&A box at the bottom of the screen to type your questions during the talks. I'm excited to introduce our first speaker today, Dr. Olya Yarychkivska. Olya completed her graduate work in the lab of Timothy Bester at Columbia, where she worked on the biology of maintenance and de novo methylation mediated by DNA methyltransferase 1. In 2019, she joined the lab of Shai Shaham at the Rockefeller for her postdoctoral training, where she began investigating the regulation of linker cell type death in C. elegans in mammals. Olya has acquired prestigious funding opportunities throughout her career, receiving both F-32 and Mosaic K-99 fellowships through the NIGMS during her postdoc. In 2024, she also received the Kellen Women's Entrepreneurship Award. Olya is on the job market this cycle and has a preprint coming out any day now related to the work she'll be telling us about today, regarding how myelin regulatory factor 1 primes a migrating cell for non-apoptotic cell death. And so with that, let's welcome Olya.
Hello, everybody. Thank you so much for the kind introduction, Austin. It is a pleasure to be here today, and today I will tell you about a novel type of non-apoptotic cell death that we work on in the Shaham lab. So cell death is an extremely important process, and it's probably...
Did we lose? I think we might have lost you. We'll give her a few more seconds to try to get back in, and then we might go ahead and move on to our second speaker. Okay, I'll go ahead and pass it to Heather.
Okay, hopefully Olya can get back in and we can see the rest of her very interesting talk. But for now, it's my pleasure to introduce who was supposed to be our second speaker, but is now our first speaker, Dr. Tony Galenza. He's an accomplished postdoctoral fellow in the lab of Dr. Lucy Erin O'Brien at Stanford School of Medicine. Tony earned his PhD in immunology from the University of Alberta under the mentorship of Dr. Aidan Foley, where he studied how the intestinal barrier mediates the lifespan extending effects of glucose and Drosophila. During his postdoc, Tony's research has focused on uncovering how stem cell progeny in the Drosophila midgut successfully integrate into the adult functional tissue without compromising barrier integrity during turnover. As part of this work, he published a beautiful paper in Nature Cell Biology demonstrating a novel mechanism of cell integration that coordinates junctional remodeling with basal stem cell terminal differentiation. Tony has also led the intestinal annotation team for the Fly Cell Atlas as a postdoc, and he's been granted multiple competitive awards, including the Canada Natural Sciences and Engineering Research Council Doctoral Postgraduate Scholarship and the CIHR Postdoctoral Fellowship, underscoring the broader significance of his work. Like Olya, Tony is also on the faculty job market this year, so stay tuned for work from the future Galenza lab. In today's talk, Tony will share a new chapter of the Drosophila intestinal differentiation story about mechanisms that promote the formation of the apical membrane, which is a key barrier forming structure. Please join me in welcoming Tony.
Thank you, Heather, for that kind introduction, and thank you to the organizers for this opportunity. Today, I'll talk about how barrier epithelia solve a fundamental problem: how to replace cells without breaking the barrier. Barrier epithelia separate us from the outside world, lining our skin, lungs, glands, and entire digestive system. This architecture is highly conserved, from vertebrates to invertebrates like the fly midgut shown here. Often, this barrier is just a single layer of cells standing between the sterile interior and the outside world full of pathogens, toxins, and other threats. To maintain barrier function, these tissues must constantly renew themselves. Like a brick wall protecting you from the harsh Bay Area weather, bricks will fall out and must be replaced. And the same is true in many of our tissues. Cells are continually lost and must be regenerated. We've learned a lot about the signals that sense damage and activate stem cells to respond, but we know much less about the stem cell progeny, the replacement cells that must actually integrate without breaking the barrier. How do these differentiating cells execute integration? This is what I care about. And I study this process in the adult Drosophila midgut. The system offers key advantages: genetic tractability, simple architecture, and the ability to image at high resolution in fixed or living tissue. Several features are shown here that are important for this talk. The brush border in green, this is the microvilli lined apical surface facing the lumen. Tight junctions in purple that seal cells together to form a leak-proof barrier. And finally, in pink, you can see these differentiating cells that I'm most interested in. These cells are called enteroblasts. They're stem cell progeny that sit basally below and between the mature enterocytes that largely compose the midgut. Early enteroblasts lack the key barrier components that they'll need as mature cells. During differentiation, they must polarize, form junctions with neighbors, and assemble their brush border all de novo. And how they accomplished this integration was unknown. And this challenge is not unique to the fly gut. Other simple adult epithelia have basal stem cells, such as the mammalian trachea, cornea, or prostate, and they have to solve the same problem. So how does this integration happen? When I started my postdoc, it was thought that new cells in the fly gut integrated through a mechanism called radial intercalation. And this mechanism was originally described in developing xenopus skin and since observed in other developmental contexts. Here, new cells extend apically and intercalate between their neighbors, forming junctions as they integrate. But this is not what we see in the adult fly gut. Instead, our lab and the lab of Daniel St. Johnston independently discovered something unexpected. Enteroblasts build their brush border before integrating in a specialized compartment we call the pre-assembled apical compartment, or PAC. In this mechanism, early enteroblasts polarize and brush border markers begin to accumulate apically. As the cell grows, the developing brush border expands into the PAC. It's an intercellular space protected from the harsh luminal environment. Once assembly is complete, the PAC then opens up to the lumen and the pre-built brush border integrates into the epithelial surface. This raised the next question: what mechanisms actually regulate PAC formation? Using electron microscopy, we were able to reveal the remarkable structure of the PAC. Here you can see a differentiating enteroblast shown in pink, and you can appreciate that it's forming these extensive junctions here in purple with its larger neighbor enterocytes. But if we focus on the PAC itself, you can really appreciate how extraordinarily complex this membrane topology actually is. The PAC contains densely packed, highly curved, developing microvilli. And again, I wanted to understand what molecular mechanisms are enabling the formation of this structure. Work in other systems has shown that a family of proteins called bar domain proteins are required for proper microvilli formation and maintenance. Some examples have been shown to localize to microvilli tips, while others organize the base. And the structure of their bar domain is the key for their membrane bending role. Bar domain proteins use their intrinsic curvature to both sense and shape membranes. This ability to sculpt curved membranes makes them excellent candidates for organizing the highly curved pack. Flies have 24 annotated bar domain proteins, but to identify which ones may be involved in pack formation, I first asked whether they're actually expressed in the differentiating cells in the midgut. And using the Fly Cell Atlas, a single cell sequencing resource where I led the intestinal annotation, we can examine expression across cell types. If we focus on the midgut epithelial lineage, we can then see which bar domain proteins are actually expressed in the cells as they differentiate. And this allowed us to narrow down the candidates to 16 proteins. I then wanted to screen these using RNAi knockdown to test if they're actually required for pack formation. However, there's a problem. In a healthy, normal gut, the cell turnover rate can actually be quite variable, and it occurs asynchronously. So this makes seeing knockdown effects hard to detect. So to get around this, I first developed a synchronized differentiation assay. And I developed this assay using a transcription factor, SOX21A. The overexpression of SOX21A in progenitors induces enteroblast differentiation across the midgut. This graph here shows the distribution of differentiating cells across stages. And what you're seeing right now is day 0, right after adult emergence. At this point, nearly all the differentiating cells are in their early stages. But after 1 day of SOX21A expression, you can see the cells have progressed synchronously as a cohort. And you can see that most are now moving into stage 2. And after another day, cells have progressed further into the later stages, forming packs and now integrating into the tissue. So with this synchronized differentiation, we can now readily detect phenotypes from RNAi knockdown. With the help of Vibhu Guru, an undergrad in my lab, we knocked down each candidate protein and assessed the impacts on differentiation and brush border formation. We screened all 16 candidates with RNAi knockdown. This graph here shows nuclear volume, as these cells will undergo several rounds of endoreplication. So this is a measure of their differentiation state, where the larger volume generally correlates with later stages. First, here I'm showing the control. The nuclei are growing as expected. And in this representative cell on the left here, you can see a large round pack forming properly, shown by the phosphomucin staining. Most of the candidates we tested similarly had no effect, but 2 showed clear defects. When we knock down endophyllin A or endo-A, we block differentiation. The cells remain small with their smaller nuclear size quantified on the right, and there's no brush border formation. Notice the lack of green phosphomucin staining on these cells. They appear to be stuck in the early stages. Similarly, knockdown of nervous rec or nuke also block differentiation. The cells remain small with small nuclei and fail to form brush borders. So from this screen, we then identified 2 bar domain proteins that are required for differentiation, endo-A and nuke. Both these proteins have been studied primarily for their role in synaptic vesicle recycling in neurons. Here, they provide the membrane bending required to reform vesicles after neurotransmitter release. However, any function in the midgut was completely unknown. So to understand their role in pack formation, I needed to know where these proteins are localizing. And given their membrane bending activity, I predicted they'd be at the developing brush border. So I examined their localization across differentiation stages. Today, I'll focus on endo-A. First, here we're looking at an early differentiating cell. You can see the merge on top showing the brush border again in green, differentiating cell in pink, and now endo-A in blue, as well as endo-A alone here below. In this early stage, Endo-A is somewhat cytoplasmic, but enriched at the apex, and it's co-localizing with the developing brush border. As the pack develops in later stages, you can see the endo-A is localizing in a ring around it. And finally, as we get into the later stages, into mature cells where they've got their full brush border, you can see the endo-A still remains localized with the brush border. And nuke as well shows a very similar localization throughout. So this supports our model that these proteins are using their membrane bending activity to shape microvilli as they grow and function. And this work provides the first insights into the molecular regulation of pack-mediated integration. Endo-A and nuke, two bar domain proteins, localize to the developing brush border and are required for pack formation and differentiation. And interestingly, their removal arrests differentiation almost entirely, which suggests an intriguing possibility that maybe pack formation acts as some sort of morphological checkpoint. So this work begins to define the mechanism of integration during homeostatic turnover, but many questions still remain. But so far, I've been talking about homeostatic renewal, this continual maintenance of barrier tissues. But barriers also face bigger challenges. In the Bay Area, you can see this is generally the mild weather that we deal with. But where I'm from in Canada, it can actually look a lot more like this. And repairing this kind of barrier in the middle of a snowstorm might seem impossible, but barrier epithelia have to do exactly this during damage and injury. Differentiating cells must execute that same integration program I just described under much more urgent conditions. And I call this emergency differentiation. When this process fails, barrier breakdown can lead to sepsis and death. How do tissues accelerate differentiation during emergency regeneration? What changes at the molecular, cellular, and tissue level? And what do cells sacrifice for that speed? And this is what I'll study in my own lab. My colleagues in the O'Brien lab, Aaron Sanders and Miriam Sun, have shown that differentiation is dramatically accelerated after injury. They tracked individual cells using overnight live imaging, measuring expression of a differentiation marker. And this marker is expressed during differentiation and then turned off once the cell matures. And in a healthy gut, as we saw earlier, this can take over 48 hours. Here you can see in this imaging time window, the marker steadily increasing over the 20 hours. But after feeding the flies a toxin to induce widespread injury in the gut, cells reach that same level in half the time. And they complete differentiation within the imaging window. So injury accelerates this process. But what does this faster differentiation actually look like? During homeostasis, differentiating cells form a single, well-organized pack. You can see here from both a side view and a top-down view, showing this really nice organized structure. But after injury, it looks a lot different. Cells cluster together and they form what I call multi-packs, these multiple small, disorganized pack-like structures. And these appear to still be intercellular compartments like packs, but they're much more fragmented. And I hope you can appreciate this striking difference in both the views compared to the control. This suggests that injury doesn't just accelerate differentiation, but it's fundamentally reorganizing how the cells are integrating. So in my lab, I want to address emergency differentiation at 3 complementary scales. At the molecular scale, what programs enable emergency differentiation, and how do they compare to healthy guts? I'll use multiomics to define these programs. At the cellular scale, how do cells coordinate emergency integration? I'll use live imaging with precision injury control. And finally, at the tissue scale, what's actually being sacrificed for speed? I'll examine the long-term costs using repeated injury. But today I want to focus on the cellular approach. To understand cellular dynamics during emergency differentiation, I need to visualize this process in real time. For this, I use the belly mount technique developed in our lab, which enables live imaging of the midgut in intact flies. This approach gives you a really beautiful view inside of the fly, you can see here. And if we zoom in, you can see the brush border really well in individual cells. And I've since extended this method for overnight imaging so that we can capture regenerative timescales. So here's an example of overnight imaging, this one using the SOX21A synchronization system I described earlier. And just remember, this is intravital imaging, so you'll see a lot of organs and structures as well, but the section of the midgut you can see is marked by this dotted line. And I'll just play a few hours kind of looping of this, but you can see a wave of these differentiating cells in pink as they grow together and form packs. But to track this process a little more clearly in detail, we can follow the individual cells. So here I've tracked a single differentiating cell over 15 hours, and you can see the pack within this cell right in the middle growing steadily. And over this time, it's growing about 4-fold increase in diameter as the cell is differentiating. So this dynamic information will really be essential to understand how injury is altering this process. But to really study the dynamics of injury, I need one more capability, the precision control over damage. And for this, I've developed a 2-photon laser ablation to kill single cells in the intact fly gut. So here you're seeing a healthy enterocyte. The nuclei are in gray, and the caspase death marker is shown in green. And I can precisely target this cell. And then 30 minutes later, after laser ablation, you can see the targeted cell is dying. Its nucleus is destroyed and it's filled with caspase marker, but the surrounding cells are completely untouched. So together with belly mount imaging, this platform enables single cell precision in a deep internal tissue of a living animal. And for the first time, I can create defined injuries and watch the complete regenerative response in real time. So barrier epithelia must perform both these maintenance and emergency regeneration. And unlike this brick house, these tissues can do this intrinsically, all on their own. My research program will define how tissues execute emergency differentiation and what they sacrifice to do so. Using live imaging, precision injury, and multi-scale analysis, I hope to reveal the molecular, cellular, and tissue-level mechanisms that underlie this fundamental challenge. And finally, I just like to thank my advisor, Lucy, and everyone in the O'Brien lab, especially Vibhu. I'll thank you all for your attention, and I'm happy to take any questions.
Wow, perfectly on time. I didn't even have to warn you. That was a beautiful talk with incredible imaging. We already have a question in the Q&A, so I'll go ahead and read that off. And while I'm reading this question, everybody else feel free to collect your thoughts and put them in the Q&A box. That you will not be able to ask the question yourself. So Chris Wright asks, do the brush border microvilli have these tip-link protein-protein bonds for structural robustness that are found in mammalian brush border in the intestine?
Oh, that's a good question. Yeah, it's known in mammals that especially in the mature brush border, to give them that really nice, tight, dense packing that they have these intermicrovilli proteins. There's not really much study that's actually happened in the fly gut, so I assume they probably do have. They share a lot of the other similar microvilli proteins, but no one specifically looked at that yet. So, yeah.
Okay. Marcia Lucas asks, how long can the flies live after a belly mount?
So yeah, the original use of the belly mount was actually so that we could belly mount image the fly and then take them off of the belly mount and let them live and then do repeated belly mounts over days or weeks. So if you do it carefully, you can actually, the flies can live. But yeah, if I do it in the overnight, longer term one, it's a little harsher on them. So it's probably not, it's probably much more difficult for them. But yeah, I think if we're careful about the technique and importantly, how we're treating them while they're on the mount. Yeah.
Okay. Raj Logan asks, what kind of remodeling or adjustments take place in the adjacent cells to allow expansion of the pack?
Yeah, that's a great question. We haven't directly looked at this, but you can definitely see as you're looking at these later stages that the neighbor enterocytes do have, they are morphologically different. You can see they are kind of bending down towards the growing cell. So we're very interested in what's going on with these neighbor enterocytes. But we haven't really specifically looked at this too much yet. Yeah.
So we have another question from Michael Layden in the panelist chat. He asks, what does the mature pack have the same area as the apical surface of the mature cell? And did you see any phenotypes that looked like differentiation occurred, but the membrane surface area was just too small?
Oh, interesting. Yeah, that's a good question. Yeah, I guess what we define as the pack kind of covers quite a few or a long stage. So you can see it start pretty small and then watch as it grows pretty much quite large. So it eventually does become basically the full apical surface of the cell. And sorry, what was the second part of that?
The second part of the question is, did you see any phenotypes that looked like differentiation occurred, but the membrane surface area was just too small?
Interesting. I would say not in these particular knockdowns. It is possible other interventions that we've done in our lab, like knocking down tori, I believe, you can see the cells still growing and seeming to differentiate, but not actually integrating. In the cases where I've been knocking down these bar domain proteins, so the cells aren't really growing. There probably are some different mechanisms separating the actual growth of the cell from the integration of the cell that we haven't figured out yet.
Okay, we have one last question before we turn everything back to Olya. Sophie Song asks, did you notice any remarkable defects during the accelerated differentiation?
During injury, you mean?
Yes, it sounds like that's what she means.
Yeah, I guess during injury, I mean, the whole gut looks really messy and bad. So it seems like there's lots of defects. But I've done kind of, again, a repeated belly mount where I injure and then do regeneration and look later. And you can have these really messy looking injury responses. And then you look a couple of days later and the gut appears to be quite normal. So it's hard to say what's actually defective about that and what, that's kind of something I'm hoping to, I think we'll need higher resolution imaging to actually figure that out.
Okay, thank you so much, Tony. And now I'm going to turn things back over to Austin.
Hi, everyone. So let's go ahead and reintroduce Olya, who is struggling with some technical difficulties, but she's back and ready to go ahead with her talk. So let's welcome her back to the seminar.
Hi, everybody. Sorry for the difficulties, but I hope, maybe it's fitting that the cell death talk is at the end of this seminar. So let me tell you today about the novel type of cell death that we study in the Shaham lab. So overall, cell death is extremely important, and it's one of the most dramatic cell behaviors, as you can see by the cell here in the middle that is dying in a very interesting way. So cell death is actually really prevalent during normal development. Up to 50% of neurons normally die. And also cell death accompanies many disease states such as neurodegeneration, cardiomyopathy, stroke, other states, and also is perturbed in cancer. So understanding cell death could really lead to novel therapeutics for many diseases. And while apoptotic cell death is the most known type of cell death, it actually doesn't explain all of the massive developmental cell death that happens normally in development. And that's because, for example, some mice that carry mutations in key apoptotic genes, as you can see here, actually can survive to adulthood and look quite normal. So this suggests that there is an alternative mechanism, a non-apoptotic mechanism, that accomplishes all of this developmental cell death that takes place. So in the Shaham lab, we are studying this novel type of cell death of the single green migrating cell that initiates its own death. This cell is born only in the male C. elegans during the early larval development. It sets off on this complex migration trajectory, and it leads the development and morphogenesis of the gonad. And as the cell arrives to the tail during the larva to adult transition, it actually initiates its own death, because if it doesn't, it sort of clogs the formation of the sperm exit channel. So this type of cell death is extremely biologically important to leave these male animals fertile. And we call this type of cell death linker cell type death. So I told you this is non-apoptotic cell death. It has non-apoptotic morphology. During migration, the cell has normal and rounded nucleus, but as the cell initiates death, it acquires these extreme nuclear deformations. And by electron microscopy, we can see these very nicely here. We can also observe the loss of peripheral heterochromatin and swelling of the organelles. And this looks completely different from the apoptotic corpse in C. elegans. Similarly, the length of this process is actually an order of magnitude higher than apoptotic process. And as the linker cell acquires these nuclear deformations, it goes through this budding of the cytoplasmic fragment that it then splits off, and then it's rounded, and then eventually it's disintegrated. So what controls the genetic pathway? Genetics is also very different. We have these key upstream regulators that contains the Wnt signaling pathways that tell the cell where to die, the timing pathway that tells the cell when to die, and the MAP kinase pathway. All of these converge on the heat shock master regulator HSF1. And shockingly, HSF1 functions non-canonically here. Normally, it's important for heat shock, for any kind of stress, to lead the animal to the cell to survival. However, here it's important to promote linker cell death. And it does so by directly upregulating the ubiquitin proteasome system, which degrades yet unknown targets to bring about this type of cell death. Interestingly, a number of these proteins that are involved in linker cell death are implicated in cancer and in neurodegeneration. And the morphology of this dying cell is also seen and conserved in various conditions of degeneration or death, both in physiological and pathological conditions. So this suggests to us this type of cell death is quite prevalent in mammals during development and in disease. It's just completely untapped. So we are using C. elegans to understand this better. And in the lab, we have previously characterized different types of mutants, these LCD initiation mutants that block the entrance to the cell death, and then these degradation mutants. I have one preprint on a new type of mutants. I characterized these splitting and engulfment mutants. that you can read. But today I will talk about the LCD priming mutants and new kinds of mutants we discovered. So how did we get here? I was really interested that this cell has two fates, both migration and death. And I was interested to understand what governs this transition from migration to death. And you can imagine as the cell migrating, it probably has all of these migration genes upregulated, but as it nears the end of its migration, it will start downregulating them. Similarly, and as it starts the death process, it will upregulate the dying genes. So we wanted to understand in a comprehensive way what exactly happens here. And to do this, I decided to perform single-cell RNA sequencing on this linker cell, which is a very rare type of cell. It's a 1 cell out of 1,000 in the male C. elegans. So normally in the population, there's less than 1% of males, and we use a special genetic mutation to increase the number of males to 30%. So for each male, we have 2 hermaphrodites, but the reporters we use to label the linker cells, we also label the sister cells in the hermaphrodite, which are called the distal tip cells. And so the ratio of the cells we get at the end is 0.03% of these linker cells in the population. So this is the workflow of the experiment. I grew a lot of worms, synchronized them, timed them to be in these phases where the cell is still migrating in this posterior leg, or it's already at the tail and has initiated the death process. I dissociated these cells and performed FACS sorting to isolate the linker cells and the distal tip cells, and then sent this for 10x genomics. And we use several different strains with several different markers. We have different replicates, so all of this data is coming together from all of these strains. And we got many different clusters and cell types. It's proved that it's actually very difficult to purify the linker cell, which sits really in the middle of the body. But we succeeded at getting this linker cell cluster over here. We confirmed its identity by looking for the expression of known linker cell death regulators on male-specific genes. And when we zoom in on this cluster, and also additionally, we confirmed that it's the linker cell cluster by comparing its unique transcription program to all the other clusters, and especially to the distal tip cells. And then when we zoom in on this cluster, we could perform pseudotime analysis, and we saw this one nice progression for transcriptional changes, which was exciting. And we used different in vivo reporters, validation, looking at the genes that we know are expressed in migrating or dying cells to really show that this is the trajectory of transcriptional changes from migrating to dying cells. And we discovered, additionally, I'm showing some novel markers like FLH2 of these specifically dying cells here. We performed additional analysis and identified these 2 subclusters for migrating and dying cells. And now we could really ask the question, what genes are differentially expressed between these subclusters, and also which genes are changing on the pseudotime trajectory. And this is an example of showing you some of the genes that are changing. For example, we saw the patterns where the genes are increasing in the dying cells up here, or they are higher up in the migrating cells and decreasing in the dying cells. We were really interested in these genes that are increasing or turning on in dying cells, as we hypothesized that those genes might be the novel regulators of linker cell death. And so we pulled all of them together from our 2 types of analyses. Nicely with the gene ontology, we were able to validate that the proteasome genes are enriched, and this is something we know. A proteasome is a regulator of linker cell death. We previously know that it comes up, increases in the dying cells, and we identified several others, other families of genes, and in total we have 252 upregulated genes. But then we needed a way to functionally test these candidates. And since most of these genes that we identified are actually essential genes, and probably we would have not found them in the previous genetic screens that we have done, we needed a way to do this specifically in the linker cell. And so we are able to do a linker cell specific RNAi. And I screened only about 10% of these genes and looked in the adult males where the cell is still present. In the wild type, the cell should be gone, but in the mutants that regulate linker cell death, the cell should still be there. And I pulled out 9 novel candidates from this screen, and we decided to focus on 2 new hits, 2 genes called MERF1 and PAN1. So recently, these 2 genes have been shown to function in the synaptic remodeling in the DD neurons in C. elegans, where PAN1 recruits MERF1, which moves to the plasma membrane. It trimerizes, it autocleaves itself, and the N-terminus goes into the nucleus and acts as a transcription factor. And the C. elegans MERF1 is really conserved. It looks very similar to a human MERF1. And the human gene is implicated in a myriad of different developmental disorders. So we were excited that we found this novel role for MERF, which brings a potential question that maybe some of these developmental disorders in humans are due to impaired normal developmental cell death. And so we needed a way to, again, study this gene in a linker cell specific way. And we developed the auxin-inducible degradation system, specifically in the linker cell, by expressing the TIR1 protein just in the linker cell. And we used 2 different tags to tag the MERF1 protein with 2 different fluorescent tags and the AID Dagron tag on 2 different sides of the N-terminus. As the MERF1 gets processed and autocleaves, the N-terminus will retain the fluorescent tags and the Dagron, so we can track it in vivo and we can destroy these N-terminus transcription factors. And upon addition of auxin, this whole E2 complex comes together. TIR1 is able to bind through auxin and initiate the polyubiquitination of this Dagron sequence and target the whole thing for proteasomal degradation. So we decided to do a series of experiments, specifically degrading MERF1 in specific developmental time points. Firstly, we wanted to confirm the phenotype and perform the depletion of MERF1 during the entire larval development. And indeed, we got a strong phenotype when the animals were on auxin, confirming our previous RNAi results. Then we reasoned, since we found this gene changing in the later phase of development, that maybe depleting it at the later phase should give us the phenotype. So we performed this experiment of putting the animals on auxin in the later phases of development. And very surprisingly, we actually did not see a significant defect. So we decided to do a reversible early auxin experiment where we placed the animals during early development on auxin, but then transferred them back to water. And interestingly, this phenotype was completely recapitulated in this condition. These cells where we depleted MERF1 had normal migration and also had this morphology that looked very much like they were still in the earlier stage of development. However, the migration program was not perturbed. And by electron microscopy, we could also observe normal nuclear and organelle morphology. So from this auxin experiment, we interestingly discovered that MERF1 acts much earlier during the larval development that we thought, and that it commits somehow the linker cell to death many hours before the death process actually initiates. So what happens to the linker cell during this time? We decided to, the cell is making this posterior turn, and we used this allele where MIRF1 was tagged with GFP and decided to follow its dynamics in vivo. So as the cell is moving dorsally, we could see that MIRF1 is localized to the plasma membrane here. However, as the cell completes this turn, we observed that MIRF1 now goes into the nucleus. So what we saw is that in vivo dynamics, where MIRF1 is accumulating in the plasma membrane, probably gets processed there, and then translocates into the nucleus, exactly at that time point where we see that auxin depletion is functionally important. And this suggests that MIRF1 is functioning way earlier and earlier than many of the other genes that we have known in this genetic pathway. And indeed, when we put together this MIRF1 depletion with the gain of function allele of HSF1, which promotes cell death in all of these upstream mutants, we also can rescue partially the cell death, suggesting that MIRF1 functions upstream of the genetic pathway. And through work with our collaborators, we found that the putative MIRF1 binding motifs are found in many of the known linker cell death regulators. They also performed the ChIP-seq experiment, and which shows that MIRF1 indeed binds to a number of known linker cell death controlling genes. And we decided to focus on one of them, this gene PQN41. This is an interesting gene because it's a polyglutamine gene. And given the similarity of the morphology of linker cell death to polyglutamine disease and Huntington's disease and that neurodegeneration, this was an interesting finding. We decided to to genetically delete this region where MIRF1 binds to PQN41 and has two putative domains, motifs that we found in here. So we created this allele using CRISPR-Cas9 and then assayed whether linker cell death is still important or perturbed here. And indeed, in this allele, we found that linker cell death is perturbed, suggesting that potentially this binding is functionally important. And additionally, we tagged PQN41 with MK2, and upon MIRF1 depletion, we observed that this signal of expression of PQN41 goes down. So this together suggests that PQN41 is a target of MIRF1.
1 minute warning.
So putting all of this data together, I want to put forward this idea that MIRF1 primes the early migrating linker cell for death. This priming happens when MIRF1 translocates into the nucleus, acts as a transcription factor, upregulates the transcription of PQN41 and possibly a number of other linker cell death genes that have enough time to be made and translated for the initiation of linker cell death. However, this priming mechanism happens more than 15 hours before the initiation of death. And this kind of priming for cell death has never been shown before. So we are really interested to explore this further. And so we have this data set. I just want to give you a taste of what's in the future for the linker cell death. We have a number of genes. We have a number of novel LCD regulators that we can functionally test in C. elegans, use it as a platform to discover genes and the mechanisms of these genes. And we want to investigate more what this priming mechanism is doing. Additionally, we have all of the genes we discovered from the migrating cells. So in the future, we could also study the motility genes that are important for migration. And in the collaborative work I'm not showing today, we have defined a first LCD biomarker by screening polyglutamine mouse brains of Huntington's disease. And so we are really interested to continue this work of transforming this knowledge we gained of linker cell death in C. elegans to a mammalian system with hope of applying it to human disease in the future. So with that, I would like to thank my mentor, Dr. Shai Shaham, and my funding sources and the resource centers for help. Thank you all.
Thanks so much, Olya. That's a really fantastic work, and so cool that you can get all the way down to profiling this one cell in the whole animal. I'm sure that was very difficult. We'll go ahead and open up the QA section to any questions. Go ahead and type. Okay, we have one question from Carrie Adler. Is the migration required to disrupt nuclear morphology? In other words, does the nucleus shear from forces encountered as the cell moves posteriorly?
That's a great question. We don't know the answer to this question yet. We do have a mutant where the linker cell is stuck more in the interior of the animal, and we do observe that it initiates the linker cell death at the appropriate time point. It is a little bit delayed, the process, and it does go through all of these stages. However, we haven't looked very carefully at the nucleus, so it would be maybe interesting to see in that mutant if this happens. But mostly the nucleus looks normal during development, during migration. I'm sure it has to be pretty flexible for this migration as the cell is squeezing through and forging its path forward. These nuclear deformations and heterochromatin change are also very interesting to me, and I have a number of different reporters, and I would like to investigate this further as well.
All right, we have a comment from Anna Scopes. She says, beautiful talks. And we have another question from Gopal Nirala. It seems linker cells migrate directionally. Is it true, or do they migrate randomly in any direction? Did you look at directional coefficient and migration speed? And is there any morphology change like EMT during migration before they die?
So they do migrate directionally. The specific turns that they make when they go dorsally and then they move posteriorly and actually move ventral posterior. Again, it seems to be a very stereotyped migration. They do have changes in their morphology during the migration, specifically sort of maybe by mid-larval stages, and the Sternberg lab has beautifully investigated that and characterized the changes in morphology of the linker cell and the changes in speed of its migration, that the speed actually increases in the later larval migration. I think it would be very interesting to understand more about this. And I'm excited that I have all this data set of the migrating genes. Hopefully there's something interesting among those that we will discover.
Thanks. We have one more question from Peng Huang. Nice talk. What is the signal that triggers the nuclear translocation during the turn?
The translocation of MRF1 to the nucleus. So in collaboration with Chris Hamill's lab, they have shown that MRF1 has this oscillation transcription, oscillating expression at the different transitions during the larval development. So it seems to be that you express MRF1, it's recruited to the cell membrane by this other gene PAN1. We do see the phenotype with PAN1. I just haven't focused on it in this talk. and it's known to recruit MIRF1 to the plasma membrane. What recruits it to the nucleus actually is really not known. We know that it autocleaves itself. It has this very ancient ICE domain that is found in the bacteriophage that is able to trimerize and cleave itself. So it's a very interesting protein. But I hope that by performing some targeted RNAi screens based on the genes that we found expressed here in our data sets, we will discover some of these partners that might be important for each step of MIRF1 processing.
Great, thanks so much, Olya. We're getting close to time. I'll ask one more really quick question, and you may have answered this. But what's kind of the function of doing a behavior like this for a cell? Like, why spend the energy to produce the cell and migrate it far away and then have it die? And sorry if you kind of addressed that, but is there some kind of signal, cells might receive in that terminal environment from having the cell die there or something like that?
Yeah, so this specific cell performs this dual function, right? As it migrates, it extends the gonad, and the gonad morphogenesis completely depends on the migration of the proper migration of the cell. But you are right about the signals. There are definitely signals that are coming from those cells that are sitting at the tail of the animal. I didn't talk today at all, but there's fascinating dynamic. There are specialized engulfing cells that are waiting there for the linker cell. And in my other preprint, we discovered this non-apoptotic role of a caspase that somehow induces the cell-cell communication between these cells and make sure that the engulfment is happening properly.
Very cool.
But just to add quickly, also in development, there's really a lot of migration that happens on top of a lot of death that happens. I wonder if these two processes can be somehow connected. This is something we will pursue in the future.
Awesome. Thank you so much, Olya. I'll go ahead and turn back over to Heather to close us out and introduce our speakers for next.
All right, thank you, Olya and Tony, for your amazing talks. The seminar has been recorded and will be available on the SDB website next week. Please join us for next month's seminar on Friday, December 12th, when Wei-Yi Qian from the New York University Grossman School of Medicine and Robin Allen from the University of Kentucky will present. Thank you all for coming.